Hello everybody, and thank you for joining us today. I'm Craig Godfrey, the website editor of Microscopy and Analysis, and I'm here to introduce this webinar entitled 3D Materials Science, a dynamic research arena any way you slice it. Today's webinar is brought to you by ZEISS Microscopy, and we're pleased to have Dr. Arno Merkel and Dr. Fabian Perez-Willard joining us today as your presenters. Dr. Merkel oversees correlative imaging and X-ray microscopy for the materials science market segment at ZEISS. He received his PhD in materials science and engineering from Northwest University in 2007, a BA in physics from Gustavus Adolphus College, and is most interested in pursuing strategies to address multi-scale research challenges with an ensemble of microscopy techniques. Dr. Perez-Willard joined ZEISS Microscopy in 2006 as an application specialist for FIB-SEM and helium-ion microscopes. At ZEISS, he moved to product management in 2011 before becoming part of the materials science market segment team in 2014. He completed his PhD in low-temperature physics at the University of Karlsruhe in Germany in 2003, where he then stayed on as the head of a multi-user interdisciplinary lab facility with a focus on the preparation of functional nanostructures by lithography and their characterization by electron microscopy before taking his talents and experience to ZEISS. Just before we begin with today's event, let me remind everyone that this is an interactive web seminar, and so you can ask questions at any time during the presentation by clicking into the questions box. Please note, only our presenters can see your questions, so don't be too shy about asking them. We will be handling a live Q&A session at the end of this 45-minute presentation as time permits. If Arno and Fabian aren't able to get to your question during this live broadcast, then they will be able to follow up with you later offline. So without further ado, let me hand the floor over to Arno Merkel, who will begin our webinar today. Okay, Arno, it's over to you.
Great, Craig, thank you very much. And welcome, everyone. My name is Arno Merkel, as Craig mentioned. Together with my colleague Fabian, we'll be running through a variety of techniques focused on strategies to obtain information in 3 dimensions, specifically for material science applications. And to start off, I think making a few comments at the beginning, while this is a bit of an introductory seminar that does focus on a variety of different techniques, we will review a couple of specific studies in the areas of alloys, composites, and a few other areas as well. However, perhaps in future webinars and future dates, we'll be able to dive in much more detail in some specific studies. The purpose really today is to give an overview and raise some awareness of really the diversity of techniques using multiple probes, whether it's light microscopy, X-ray microscopy, electron microscopy, or ion microscopy, are ways in which it's becoming easier and easier, more routine every day to obtain 3-dimensional information for microstructural investigation. And toward the end of this talk, I'll come back and discuss how experiments are now being designed really to connect multiple length scales and are no longer becoming a heroic effort, but a much more routine effort. So before I go any further, I'd like to, the work is really a combined effort from a number of collaborators, also some not listed on this list here, but we will be showing a number of examples from a variety of groups around the world, including UCL and University of Manchester, Arizona State University, IIT, Argonne National Lab, and others, DTU, as well as our partners, Xradia, working on some of the grain orientation crystallography reconstruction packages. I'd like to mention up front that it couldn't be done without the help of some collaborators and scientific partners on that front. Now to move into some of the technical content, here's a display of, we'll be touching on a few different applications today. But the 3D material science area is really blossoming. You do see the emergence of a number of smaller conferences that are dedicated to this topic, and really with the appreciation of now tackling 3D microstructure characterization, but also quantification and modeling on a variety of types of materials. So we see coming in and out of our lab every day a variety of materials, including polymers or coatings or ceramic, composite, and we'll touch on a few of these in a little more detail, but it's quite a diverse field and quite a dynamic field. And in particular, the ability to look at these types of structures in their natural environments is something that is really quite a trend. So looking at samples not only in their static state, but perhaps those that have undergone some kind of microstructural evolution is really the driving force behind better understanding of mechanisms and also future design of materials that perform in a more suitable way, or a way in which they were intended to. Now, to take a little bit closer look at length scales, maybe foreshadowing to the end of the presentation, as I mentioned, looking at in situ studies, for example, this is a bit of corrosion. This is a couple millimeter sized magnesium alloy, which has been subject to multiple stages of corrosion and imaged with an X-ray microscope, in this case the Versa X-ray microscope. And this was imaged actually under a number of different conditions over time to really halt the corrosion process at a particular spot. Subsequently, regions of interest were located here for both further nanoscale XRM imaging and subsequent FIB-SEM tomography imaging. And really the trick here is in combining this three-dimensional data into a cohesive model as well, so it can be analyzed appropriately. But here we really have the tools now today to perform such targeted and efficient 3D characterization steps across multiple scales. We'll have a little bit more about, much more detail about this type of experiment coming up at the end. Now, looking at some length scales that we'll cover today, I've depicted on this graph the X-ray and electron and ion techniques related to 3D material science. Light microscopy is indicated here very simplistically, although it covers a large swath of this left-hand side of this graph here, really depending on the exact type of technique. But to keep things simple on this graph, I wanted to isolate here this connectivity of scales. It really is, we have a portfolio, and the world really has developed, and researchers have developed a number of techniques that are increasing in their overlap with one another, which is a very good news for tackling some scientific challenges in the 3D material science community. So we're going to march down the chain here, beginning with the light microscopy, and then working our way down toward X-ray microscopy and FIB-SEM tomography, and then exploring how we link these together. Now this plots sample size versus 3D voxel resolution, and these are approximate spaces here, all come with their different strengths and limitations and degrees of overlap and suitability. But the fact that one can now, the researcher can now really choose not simply just what is convenient for your technique that is being used, but really utilize perhaps a number of different techniques to explore a single length scale to give complementary information is really a benefit, and benefit also to the analysis. Now moving along, I'd like to simply exhibit and display the diversity of modalities and length scales and platforms that we also manufacture and develop at ZEISS, ranging from stereo light microscopy on the left-hand side, which is commonly used for routine tests or even sample mounting and experiments, all the way down through helium ion microscopy, pushing the sub-nanometer world on the right-hand side, and everything in between, including a variety of different light microscopy, X-ray microscopy, and SEM and ion microscopy techniques. So it's really the glue of putting these together and also making sense of workflows that span these, which is, we strongly believe, is the future of microscopy. Now we'll touch on a few of these platforms. First, very briefly, I'd like to review a couple of the light microscopy techniques which are being used for obtaining three-dimensional information. One of them, which many of you may be familiar with, includes serial sectioning, so where you would use a mechanical grinding and polishing process to subsequently move through a material and image it with any type of light microscope. That's been done quite routinely over the past decade or two, and is really growing in popularity also with the automated serial sectioning approaches that are emerging. So today I'd just like to focus briefly on a recent product that was highlighted and launched just a few months ago. The ZEISS LSM 800 for materials is the next in the stage of confocal microscope used for measuring a variety of different dimensions, but specifically for three-dimensional studies, really excels at performing topographic maps and related information. You can see here the diversity of modalities and contrast mechanisms that are available and accessible to you, including bright field and dark field imaging, DIC, polarization, contrast, using a variety of different filters, and here with a lambda plate. This diversity of contrast mechanisms really allows you to fully characterize a specimen, here depicted in 2 dimensions. Now, if you move on into the 3rd dimension, obviously the confocal principle, which is well known and utilized across a number of fields, notably in the life sciences area, is also getting quite a bit of use in material science for either soft material studies or measuring surfaces of samples that have a certain topography. Now, the principle utilizes the out of focus information, which is blocked by a pinhole, and as this illumination system is operated, an optical slice is then selected by scanning in X and Y. And what is then done is that you move essentially in Z to collect an image stack, and if it's a transparent material, you have the structure of the entire volume there. If it's a non-transparent material, you effectively have in focus whatever the surface is rendered. And you can reconstruct that then into a surface map where you can measure things like roughness, but even begin to segment different volumes of different features. Now here you're calculating the height for every pixel that is out of the Z stack, and here, as you can see in the lower right, a height map can be produced here at a certain sample. Looking a little bit more closely at this, what we see in terms of applications fields for this technique is quite diverse. So surface metrology, this is a scratch that's been placed into a specimen. You can quantify the volume here and the exact roughness or profile of that scratch. That could be applied to maybe a wear track or some type of a tribological investigation. Obviously, some structural analysis of a composite using different fluorescent modes can be performed, as well as layer thickness measurements. So in 3 dimensions here, the layer thickness can be measured using that same confocal technique. Also, for some porous materials, you're able to measure into pores and measure some of the porosity of these specimens. Now, the diversity of techniques and applications is quite large in the light microscopy field. To keep things relatively brief today, I'll just highlight one study here, which is where erosion on a contact here was measured. This was some wear testing on an electrical switch. And you see this curved surface here with a few different deposits or accumulated material here. As it's run through this 3-dimensional confocal imaging technique, you're then able to effectively obtain that surface roughness and shape information, which then can be measured and quantified. And so you could back out now. You have a cylindrical shape here, but that can then be backed out. You can correct for that volume by subtracting that cylinder from the surface, and then perform various measurements of the void volume or the accumulated material volume, or other quantities here that are depicted in red, blue, and then green here in the lower picture. So that gives you a sense for the 3-dimensional data that can be attained simply using the confocal principle with the new LSM 800 for materials. Now with that, I wanted to move along briefly to X-ray microscopy to give you a flavor for now a technique that operates perhaps at similar length scales to light microscopy. It uses a completely different approach, now using X-rays instead of light microscopy, and it's very much a dedicated or a system that is geared toward purely 3-dimensional and non-destructive imaging. Now, to calibrate oneself, okay, so we're looking at these shapes here in the center, the blue shapes here. We'll touch on submicron X-ray microscopy and nanoscale X-ray microscopy. the Versa and Ultra platforms respectively, and we'll touch on a few application areas that are emerging with respect to this. Now, resolution-wise, you saw in the previous graph, submicron microscopy runs the gamut from spatial resolutions down approaching half a micron, so officially 0.7 microns, up to tens of micron voxel dimensions, whereas the nanoscale area requires generally smaller fields of view, smaller specimens, but is now able to obtain resolution in the tens of nanometer spatial resolution with a minimum voxel dimension of 16 nanometers. Now, the basic technique for obtaining X-ray tomography, as many of you are likely familiar, but for those who are not, you effectively shine an X-ray source through a specimen. That is then detected on a 2D detector, in this case the Versa's optical magnification detector that couples a scintillator with a light microscope lens to a CCD detector. And as you rotate the specimen at multiple increments, it can be up to several thousand different projections, or as few as a few hundred, really depending on the resolution requirements. You obtain these radiographs that are then collected, and these are then processed, corrected, and reconstructed into a virtual 3D volume that can then be further quantified. Things like the density of the material are inherently inside of the data and can be extracted as well, although you're not given any direct compositional information from this. Spectroscopic techniques or such as EDS or fluorescence would be then more logically performed to obtain specific chemical information. The beauty of this technique is that you have that three-dimensional information non-destructively, and you can subsequently go back and perhaps deform or anneal that sample and look at its evolution. Going into a little bit more detail on the Versa length scale, again, this is the submicron X-ray microscope. Looking at a movie here, which is playing of a fiber composite sample. This is almost a millimeter in diameter here with a 0.9 micron voxel dimension that was obtained. You can clearly see individual fibers. This is actually a fiber composite that underwent tensile load, which was then fractured. Now, the ability here, this would be an interior tomography where part of the sample does extend beyond this field of view, so you can probe interior volumes at different areas. This gives you a flavor for the type of information, looking at the orientation of the different fibers, and can give you an insight into what failure mechanisms occurred. So looking at the geometry a bit more of the technique, I showed this graphic on the right-hand side and a few slides ago. And now, by incorporating this optical magnification, we've developed a system which was inspired by many synchrotron-based approaches to move beyond conventional micro-CT, which conventionally utilizes a single flat-panel detector and relies heavily on geometric magnification, which places many constraints on the performance of the source, stability, and most importantly, the sample dimension. Now, the sample dimension and size, or the environment that it's placed in, when you place it in a dual-stage magnification system, allows you great freedom in the ability to obtain high-resolution information at multiple points within the sample. So it's critical to remember that high spatial resolution is maintained as that sample is moved away from the source. Now, this could be a large, awkward-shaped sample or a flat sample that you can't afford to break up. Perhaps you have further experiments or conditions you'd like to subject that sample to, or it's a sample that you've placed inside of a chamber to look at corrosion or look at tensile failure or electrochemical stimulation and cycling. Could be a variety of things. It's really the most flexible platform for obtaining true submicron resolution across a wide spectrum of sample shapes and sizes. And the trick to doing so is effectively this coupling of detailed scintillation and highly pure and well-controlled scintillation materials with an optical objective and a CCD detector that's sitting behind here. So the X-rays are converted to optical light. They're magnified primarily by this optical mechanism, which occurs after the sample, and then onto a CCD detector. Now, to summarize some of the key enabling features and applications that are now enabled with such an architecture for an X-ray microscope, the ability to perform interior tomography, which relies on resolution at a distance, or RAD, so virtually zooming in at high resolution to the interior of samples, but also being able to do fast scout scans that give you an impression of the entire sample, and coupling those, and even automating to some degree that procedure of essentially clicking to a region and performing a high resolution at any point in the sample that the user chooses. Of course, the ability to look at samples in their natural environments in situ stages, such as this tensile stage here from Devon, which was placed here into the Versa microscope. Looking at optimized absorption contrast is a key feature, and this has to do with the efficient use of scintillation material and coupling to the optical system. A unique phase contrast, propagation phase contrast mechanism, which allows to extend another contrast mechanism beyond pure absorption, which gives you density. Phase propagation allows you to detect features which would otherwise not be detectable. And finally, something I'll touch on in a few moments, an emerging, highly unique technique known as lab DCT, or diffraction contrast tomography in the laboratory, which is an analog to EBSD in the electron microscope, where we're now able to obtain crystallographic information about the grain orientation of grains within various materials, metals or others. Now, critically, the sample dimension is of most concern in conventional micro-CT, whereas when the sample grows, it will typically require you to change the working distance so that you're now further away from the specimen on the rotation axis, which effectively degrades your ultimate resolution that you can achieve. That's circumvented in the Versa design, where you effectively have constant resolution of a micron or less over a long range of different working distances. So you're invariable to that effect. Now, moving along, I mentioned phase contrast. This is an example of an aluminum silicon alloy, which is conventionally has very little density difference between the two domains. And when you place the microscope in a phase contrast condition by moving the source and the detector away from the sample, increasing that propagation distance, the definition of the edges becomes much more clear. And even in this case, you can see some of these fine needles or plates, which are indicative of the eutectic structure within the aluminum matrix here. This technique basically adds another level another lever for the user to be able to modulate the contrast mechanism. Of course, segmentation becomes a challenge in such an image, and the community is working on ways to process such information to make it more segmentable. So better imaging data, raw data, whether it's absorption or phase contrast, leads to better segmentation ability, which is primarily what is desired to be able to then finally improve the quantitative analysis, which yield better materials models. Now, keeping it rather short, I wanted to touch on just a few recent highlights of the extension of the platform and looking at large engineering objects as well. This is a catalytic converter, and with the implementation of a flat panel extension in the Versa, you're able now to scout very large objects, over 100 millimeters in size here, but also maintain the ability here to use the other objective lenses to optically zoom into interior regions and obtain higher resolution information. That's also depicted on the following slide in a carbon fiber composite. This is a chunk of a hockey stick, which was undergoing some load and 3-point bending, and the failure at some of these joints was investigated. This is about 90 millimeters in length, 20 millimeters in height, and the flat panel extension can easily and quickly image the large specimen, but then even zooming in down with the 0.4x and 4x lens completely non-destructively without sectioning the sample, you're able to obtain resolutions down to being able to detect individual fibers within the sample, and obviously the cracks that are contained in here as well. Now, for a few minutes, I want to touch on another emerging modality which was recently developed and launched on the VERSA platform, known as laboratory diffraction contrast tomography, really with the vision to turn 3D grain mapping into a routine tool. We can, using this technique and obtaining diffraction patterns in the X-ray microscope, we can fully reconstruct this into a 3D volume that gives us the grain centers, the grain orientation, and the volume of those grains. Where traditionally in absorption contrast, you would have no contrast from a polycrystalline sample, single-phase polycrystalline sample. This unlocks the ability now to look at the effect that crystallography has on various mechanisms, either failure mechanisms like stress corrosion cracking or different failures, as well as growth, perhaps grain growth studies as well. Moving along, this shows a bit of the architecture. I don't have time to get into detail of this today, but an aperture is utilized here with a specialized detector where a number of different diffraction patterns are acquired and then reconstructed using reconstruction algorithms developed jointly with our partners at XNovo Technology to reconstruct this crystallographic data into, and these are Laue patterns, into a data set that gives you the three-dimensional, non-destructive three-dimensional grain orientation information. Now, one of the first validation experiments was done on a beta titanium alloy, and here's what you would see in a conventional absorption scan. You don't see much information other than the exterior of the specimen, but we can now populate this after a DCT analysis with the rich crystallographic data. Those cubes centers indicate the centers of each grain, the size of each grain by their size, and then the crystallographic orientation with their color. Now, this is the first step, that can then be tessellated, for instance, into a structure. But we're working on ways also to extend that morphological analysis into something that looks more EBSD-like. And we've measured this and tested these results against EBSD, against synchrotron-based laboratory diffraction contrast tomography, and they agree quite closely. Obviously, EBSD has a finer, higher resolution, which is highly complementary to performing the time-dependent, larger volume study first with X-ray microscope, and coupling that even to modeling systems or brain growth models. So it was validated against a number of techniques, and really the direction that this is going. This is courtesy of some work being done jointly at the University of Manchester, looking at the growth of these are sintered copper particles that are that underwent different heat treatments. But again, we're doing this non-destructively, so we can now explore the growth of individual grains or the statistical orientation change that occurs between different types of grains, and really populate our understanding of models to help understand what the drivers of growth here and change in the microstructure are. So that's just one example. It's full of rich quantitative information. For example, here is the grain distribution size of those copper particles and the frequency of the grain diameters. And you can see that's the initial step, followed by the final step after all of the heat treatments were done. You see a shift in this, which is to be expected that you have a number of quite large grains that have appeared at the expense of some of the smaller ones, which is expected. The details, however, can be investigated and are being investigated at the moment. So I'd like to wrap up this section. I already mentioned this slide to really highlight the resolution at a distance interior tomography, the ability to do in situ experiments, and a number of different contrast mechanisms, both absorption, phase, and lab DCT grain mapping. It's really this extension and growth of X-ray microscopy in the community. And a few highlights also from the literature of some work being done on, very nice work being done on studying the three-dimensional crack front morphology. This is in stress corrosion cracking in steel, some work done in Australia, and coupling to some light microscopy techniques of first starting with the surface, moving toward other applications in the energy materials areas, looking at entire encased batteries and studying the effects of the microstructure here on their performance also over time. And you can even image, this is an 18650 cell that was imaged now in an interior tomography was taken here on the left-hand side. And after several cycles, electrochemical cycles, cracks were witnessed on the right-hand side at specific locations. And this can be quantified as depicted here in the green circles. So look at this evolution information completely in an embedded, non-destructive way is quite a powerful technique. Now, I'd like to wrap up the X-ray microscopy section with a few comments on the net. So that was the Versa platform at the beginning. The nanostate scale technique, the ZEISS Xradia Ultra, really progresses the field beyond the submicron range properly into the nanoscale resolution range in the tens of nanometer resolution. And in doing so, it requires the use of different X-ray optics, such as condenser lenses and objective lenses. This type of information is essential to looking at pore structures, perhaps, that are on that scale of tens of microns. The table isn't showing up here, actually. This is only the large field of view that's shown here. There's a high resolution mode as well that has a voxel dimension of 16 nanometers and a field of view of 16 microns, and an ultimate 2D resolution of 50 nanometers. Now, in doing so, I'll skip through this for the sake of time, but those of you interested, we have quite a bit of literature on some of the optics that are required to manipulate the X-rays directly. This condenser, the glass capillary condensers, as well as these e-beam patterned zone plates, and details of the detection system as well. Now, I showed a battery result from the Versa just a moment ago. If you really zoom in on an individual cathode particle, this is what you would see. This is about, maybe 10 microns in diameter here. One that's a fresh particle and the other that is after 50 charge cycles. You see a dramatic difference in the interior porosity, and also some of the cracks can be detected that run to the surface and guide some of the lithiation process in these individual cathode particles of a battery. This is some work done in collaboration with Argonne National Lab and looking at the evolution of individual particles here, and possibly even coupling this to electron microscopy experiments. And my final slide on the Ultra is really highlighting some of the future directions, something that a number of groups are very active in now. Some of the very first results from last year is showing, looking at some work of, this is some indentation done on dentin, and this dentin material is now being indented or could be compressed. This is a sharp indenture diamond indenture tip, and we're looking at the crack propagation in this natural composite material, which is, you see some of the dentin tubules running down through the left here. These are a few different views in the segmented volume here on the upper right-hand side. So this helps in our understanding of how cracks propagate. These are multiple different three-dimensional tomography acquisitions at the nanoscale over time. This field of view here is 65 microns across. Stepping back a second, what I'd like to do now is actually pass along to my colleague Fabian Perez-Bullard. He'll speak briefly on FibSEM tomography, now passing down to the next length scale of characterization in 3 dimensions, which overlaps with nanoscale X-ray tomography but takes it properly into the single nanometer range. So Fabian, over to you, and I'll conclude later with a final wrap-up section. It's all yours.
Thank you, Arno. As Arno has mentioned, the next topic of this presentation is the topic of FibSEM tomography. Coming back to our 3D multiscale microscopy graph, FibSEM tomography covers the application space highlighted in blue. Volumes of interest with sizes from a few microns up to around 100 microns can be analyzed with a resolution down to a few nanometers. FibSEM tomography thus fits between X-ray microscopy tomography and other tomography techniques like TM or atom probe tomography. How does FIB-SEM tomography work? Well, first of all, you need a two-beam FIB-SEM instrument or cross-beam instrument, like the one depicted here. On the top of the instrument, you can see the SEM column, and at an angle of 54 degrees, there is a FIB column mounted. The sample is in the chamber, positioned in the so-called coincidence point. The coincidence point is the point where both SEM and FIB beams intersect. In this position, the sample can be machined with a FIB with nanometer scale precision, and the process monitored with the SEM. The ZEISS crossbeam is designed in a way that the patterning process can be imaged live, as you can see right now, at the best instrument's resolution with any of the available detectors. You can see how the focused ion beam is being used to machine a spiral onto a silicon substrate. The spiral has a spiral pitch of 150 nanometers and a line width of 20 nanometers. Once you have found the region of interest, the sample is tilted towards the FIB column. Then a trench is machined into the sample so that the area or volume of interest is accessible by the SEM beam. And then you start slicing the sample using the focused ion beam and acquiring images with the SEM, as you can see here. This process is iterated a number of times. Typically, a FIB-SEM tomography run has 100 to thousands of images. And once this process is finished, you end up having a stack of images, as you can see here. The next slide shows you an example of raw data acquired on a niobium-3 tin multifilament superconducting cable. These images were acquired using a backscattered electron detector. You can see the different materials present in the sample, and you can also see the grain structure of some of the faces. Once you have acquired an image stack, the next task is to gain a 3D representation of the sample out of the raw data. 3D reconstruction, visualization, and analysis is key and is done using different software packages. And here you see an example of such a visualization done on an ISO 400 color film sample. You see a grain has been segmented of 1 micrometer in size. And this grain belongs to one of the color-sensitive layers of the sample. In this case, the voxel size is 5 nanometers. This means the voxel is 5 nanometers by 5 nanometers by 5 nanometers. Typically, the direction of advance of the focused ion beam is called the Z direction. ZEISS offers different FIB-SEM tomography solutions. For most advanced requirements, the crossbeam instrument is combined with a 3D tomography software module, which is part of the so-called Atlas 5 solutions package. As you saw already, FIB-SEM tomography is a destructive technique. Therefore, an advanced tomography solution needs to allow the user to acquire all relevant information during the tomography run, as unfortunately the experiment cannot be repeated. Because of the large number of cutting and imaging iterations involved, another important aspect is to develop techniques to optimize the throughput of the experiment. Last but not least, the ultimate goal is to achieve the best possible 3D resolution. Maximum information is achieved using a complete detection system. Here you can see a plot. The central top graph is a plot of the energy or the distribution of electrons as a function of energy. These are the electrons that emerge from the sample once the sample has been impacted by the primary beam. The different electrons, for example, in green, the so-called secondary electrons, carry different information, in this case topographical information, while the electrons in blue, the backscattered electrons, have interacted with the nuclei of the elements present in the sample, and they carry compositional information. At 1 kilovolt, as you can see, the information depth is still greater than 10 nanometers. If we now focus on the red trajectories of the backscattered electrons, you see at 1 kilovolt, using a so-called energy selective backscatter detector, it is possible to select a window of energies to only detect so-called low loss electrons. That is, backscattered electrons that have lost only a small amount of their original energy. These come from very near surface regions. In the plot, in the last plot of the slide, only trajectories of electrons that have lost a maximum of 100 electron volts of energy are shown. This is realized, as you can see on the very right image. The energy selective backscatter detector has a grid in front of the detector. The grid is biased at the voltage, in this case, of 900 volts. Therefore, only backscattered electrons with an energy greater than 900 evolts, and of course smaller than the primary energy, that is, electrons in a window of 100 evolts, can be detected. A histogram showing the number of backscattered electrons as a function of their depth shows a maximum depth of 16 nanometers in the unfiltered case, in the case where we are detecting all backscattered electrons. Now, using the filtering grid and applying a potential of 900 evolts, or potential of 900 volts, we can now select the energy window between 900 evolts and 1 kilo electron volts. And you can see now the information depth is below 2.5 nanometers. Therefore, ESB detection, energy selective backscatter detection, allows us to probe depths that are less than 3 nanometers. While the detection system, and especially the ESB detector, are integral part of the crossbeam instrument, the advanced Atlas V 3D tomography package is an option. It incorporates a number of features that are listed on this slide. You can see an intuitive, workflow-oriented user interface that allows the users to very easily set up a tomography run. It is possible to acquire multiple signals in parallel. Further, you can acquire images with a very high pixel density of up to 50K by 40K pixels. And further, it is possible to define exact regions of interest that need to be imaged. These images could have any arbitrary shape. Further, the solution exploits the possibility of simultaneously performing SEM imaging while doing the flip milling. There are autofocus and autostick routines that can be executed through the run to maintain the best image quality possible. Further, we have taken care of correcting for any drift of SEM and FIP beams and tracking the thickness of the slices. These are measured live, and there is a feedback mechanism to ensure that the system maintains a constant slice thickness through the whole tomography run. The exact region of interest feature is illustrated on this slide. The idea is to enhance the throughput of the FIPSEM tomography runs by acquiring SEM images only at the exact regions that the user is interested in. As the milling progresses, the system periodically collects keyframes of the entire field of view. These are the 3 images on the top of the figure. Using these keyframes, the user can define new regions of interest for data collection, remove existing regions of interest, or reshape them as needed without interrupting the workflow. In this way, the operator can adapt the tomography run to adjust to the features that they find in their samples. I mentioned that during 3D FIP tomography, Atlas V uses a feedback mechanism to make sure the thickness of all the slices stays consistent over the course of the tomography run. This is done by using a pattern of lines, which is shown on the left image. This pattern is produced on the top surface of the sample. And the distance between the outer lines is measured, or in the cross-section, these outer lines appeared as notches. The distance of these notches is tracked continuously during the run, and the distance change of the outer notches will, or is directly connected to the slice thickness. Further, the 3 internal lines you see on the left sketch can be used to track sample drift too. As I said before, using this information, Atlas V automatically adjusts the FIP milling and SEM image collection to make sure the tomography run proceeds at a constant rate. This has been experimentally demonstrated by researchers at NIH. They basically took a model sample, gold nanobeads, embedded in a matrix. These nanobeads are very small, 50 to 100 nanometers in diameter. And then they run an Atlas V tomography run at a resolution of 3.9 by 3.9 nanometers in XY direction and cutting slices of 5 nanometers thickness. And as you can see here on the left side, you see the original plane, the XY plane. And on the right side, you see one of the reconstructed planes, the YZ plane. And you can see both images are very similar. They show similar resolution. And this demonstrates that the resolution achieved during the FIPSEM tomography run is isotropic. The same group at NIH has pushed Atlas V even further and reported in a recent publication a consistent set slice thickness of 3 nanometers. On the left, you can see a plot out of this publication where you can see how the distance or the slice thicknesses are measured as a function of slice number. At the very beginning, the process needs some time to stabilize. It quickly stabilizes, and then it maintains a constant slice thickness of 3 nanometers through the rest of the experiment. This is also an interesting example. It shows a huge data set of a C. elegans worm. What is particularly impressive about this data set is that it contains over 10,000 sections, covering a length of more than 80 micrometers in Z direction. Not only a constant slice thickness could be achieved, but also all images are nicely focused and sticked. To finalize this section on FIPSEM tomography, just a short note on 3D analytics. Both the acquisition of 3D EDS and EBSD datasets is possible. On the left, you see an example for a 3D EDS dataset on a magnetic material. In red, you can see iron, in yellow and blue, praseodymium and oxygen. The right image shows an example of a 3D EBSD dataset. The sample is electrodeposited nanocrystalline nickel. In Atlas V, we have recently incorporated the EDS capability. Here is a dataset on lead-free solder which is courtesy of one of the very first users, Dr. Marco Cantoni from the EPFL in Lausanne, Switzerland. You can see the material contains different precipitates, and now we can add the EDS information to the tomography set. On the left movie, you can see the in-lens SE signal. The images were acquired at 1.8 kilovolts. The voxel size is 10 by 10 by 10 cubic nanometers. On the right side, you see EDS maps that were obtained from the region that has been framed with a green frame. The EDS maps were acquired only every 10th slide, because the information depth of the EDS signal is larger than the in-lens SE signal. What's important to note is that the EDS and SE images were acquired at different landing energies of the SEM. The workflow is described in more detail on this slide. So here you see the acquisition of the image N-1. The next slide would be slice number N. Now the landing energy is changed from imaging conditions to EDS conditions. In this case, we switch from 1.8 kilovolts to 6 kilovolts. And an EDS map is acquired. After acquisition of the EDS data, we change back to the imaging conditions, 1.8 kilovolts in this case, and acquire the next slice image. This process is iterated a number of times, and you end up having a 3D data stack, which can then be reconstructed. On the left side, you see the reconstruction for the in-lens SE data acquired at 1.8 kilovolt, featuring the best possible 3D resolution because the imaging was done at a low kilovoltage. And on the right, you see the EDS results. The EDS maps were acquired at 6 kilovolts. This means the landing energy was sufficient to generate the needed X-ray. The needed X-rays. After this short excursion to FIB-SEM tomography, I will now hand over to Arno Merkle, who will explain how FIB-SEM tomography and the other 3D microscopy techniques already presented in this webinar can be combined in correlative workflows to address multi-scale problems in material science.
Thank you very much, Fabian, for that nice overview of FIB-SEM tomography. I'd like to wrap up in the final few minutes of this webinar to see how we connect some of these length scales now into proper multi-scale three-dimensional workflows. And I'll do this through the context of a single example in aluminum alloys that connect submicron X-ray tomography all down through cross-beam FIB-SEM tomography using these two techniques. You saw at the very beginning I showed a technique that utilized all three of these techniques, Versa, Ultra, and cross-beam, to look at corrosion in magnesium alloys, which really exhibits multiple stages of characterization along this step. Now, this multi-scale challenge obviously spans different scales, but it does require multiple modalities as well. So as modalities, we mean different contrast mechanisms, whether it's absorption or EDS spectroscopy or polarized light microscopy. They're all helpful in one way or the other to fully characterizing the system. But multiple dimensions are often required, such as 2D is often sufficient, 3D is often desired, and even 4D gives you a real insight into the behavior of materials over time. Now, naturally, there's no magical instrument that does all of this, and it requires you to connect these in different ways, and then also scale these in an appropriate way so that multiple users that are not necessarily in a single location can contribute to a multi-scale experiment. So a big challenge both on the acquisition side but also on the analysis side. And I'll give you some insight into one specific study done with colleagues and collaborators at Arizona State University, looking into 7,000 series aluminum alloys, which are critically used in the aerospace industry, but also several other industries. And corrosion is a big concern. Obviously, these lightweight alloys that have a high strength-to-weight ratio and have reasonable cost are desirable to use, but only if corrosion and other failures can be controlled and understood. Now, when they make these alloys, typically you'll have some impurities in here, but you'll also have some other elements in here that are intentionally placed in to strengthen the alloy. So principally, you'll have the aluminum grains themselves. You'll have second phase particles, such as inclusions, which are these larger, typically iron-bearing inclusions, so these bright white specks that you see here in this middle graph. They're maybe 10, 20, 30 microns in size. And then nanoscale magnesium-rich precipitates, which are intentionally placed into the alloy to strengthen the material. Now, to characterize this, we utilize both X-ray tomography as well as FibSem tomography. First, to get here in purple, you see a distribution, purple and red, a distribution of these inclusions, so these larger scale inclusions. And then finally, on the right-hand side, we really need to zoom into the nanoscale to isolate a single inclusion. You see this bright feature is a silicon-bearing inclusion. We want to understand the distribution of the nanoscale precipitates around that area. Where are they? Are they at the grain boundaries, inside the grains? Are they in the inclusion themselves? And from this information, we can begin to build an understanding of what drives the corrosion process, what drives the crack propagation process, what needs to be present in order for that to occur. Does a certain chemistry or grain boundary interface need to be there? Does a void need to be nucleated? Lots of different variables here, which can really only be inspected by coupling multiple scales of techniques efficiently together and feeding those in appropriately to the appropriate quantification techniques. So this technique was actually employed first to scan the sample with X-ray tomography, then moved to FibSem tomography, but we didn't do this blindly. FibSem, as Fabian showed just a moment ago, is a fantastic and diverse technique, and it's amplified, and it's great if you know where you're looking, what you're looking for. So we basically have demonstrated over the past couple of years now a very efficient technique utilizing software known as Atlas V, which is an online direct control of the ZEISS FibSem, the crossbeam platform, to guide and automate this procedure of targeted FibSem acquisitions. You load in an X-ray data set here depicted in yellow. You then scroll down through the interface and have detected a void. So this black region in that green square is a void that I'd like to then inspect that is 10 microns below the surface. I otherwise would not have known that that feature is there. And now I can pick multiple regions and proceed with the automated 3D FibSem tomography. I've captured this inclusion and the void behind it as well. And so now I have a rich data set that I can quantify. And I'll skip forward just for the sake of time past the video and show you some of the results. This is the segmented volume now of the FibSem volume, showing in color some of the grains and all of the different inclusions. Okay, that's nice, and you can measure some of the volume fraction of the precipitates is 2.37%. But what's of most interest is really to get some statistical understanding now of where these precipitates are, and does the shape or size have an influence on where they're located with specific relation to some of the aluminum grains. So that was done for this data set, and that we're centered around this inclusion and void gives us now a symmetric environment in which we can quantify this information. The red precipitates are those that are found at the grain boundary. The blue ones are those that are found inside of the aluminum grains, and now we can statistically compare these to one another and measure, for example, that the mean volume is more than twice the size at the grain boundaries as they are on the grain interior. And this type of information can really feed models. This is expected. I think you have obviously faster diffusion along the grain boundaries, and they act as a nucleation site or faster nucleation site. So while that's expected, calibrating the exact dimension difference is important and fed in by this type of information. So that is one brief example of how we're connecting a workflow to look at multiple three-dimensional data sets. I think can be applied to many different types of material systems. Again, I wanted to thank a number of the collaborators I showed some of the work from that we've done. And with that, I think I'd like to wrap up today's session. Thank Fabian again and Craig for hosting today. I believe we'll be taking a few questions at the end. Thanks for your attention on this webinar on 3D material science and tomography techniques.